A very good afternoon to all. How are you all? Hello, Dhruvi. Hello, Priyadarshini. Hello, Chichni. So quickly answer through poll. My audio is clear and my screen is visible. So today we are going to discuss biotechnology principle and processes. So bio means living and technology means techniques. So biotechnology deals with the technique of using living organisms, okay, and the enzyme from organism to produce the product and the process useful to humans. Biotechnology has been used in various fields like agriculture, in improvement in food technology, in fermentation technology, in pharmaceuticals. So according to EFB, which is a European Federation of Biotechnology, EFB compass the traditional view as well as the modern biotechnology. And they have given the definition that biotechnology is the integration of science, organism, cells, parts, and the molecular analogs for the product and services. That means we use the living organisms to produce the product in the host cell. So there are two core principles of biotechnology. Number one is genetic engineering and the second one is bioprocess engineering. In genetic engineering, that means we are creating the recombinant DNA. So genetic engineering is the creation of rDNA. Here R represents the recombinant DNA. For example, this is a genome. And this is a gene of interest or the desirable gene. We know that the genome, they contains a lot of genes, but we are only interested in the gene of interest. So we have to extract or isolate this gene of interest. For that, we require a Caesar, right? And function of Caesar is to cut, right? So here the molecular Caesar are restriction enzymes. So I expose this gene of the genome with the restriction enzyme. So restriction enzyme will recognize the specific site. It will cut the gene of interest. And let's say this is a vector. Now I have to insert this gene of interest into the vector. So I have to cut the vector also with the same restriction enzyme. Now this is a recombinant vector because it contains the gene of interest. When this recombinant vector or the recombinant DNA is inserted into the host organism, and as the host reproduces, the number of this recombinant DNA also increases in the host cells, so that we can easily extract the product from the host cell. So this is the main role of genetic engineering. Bioprocess engineering, we also known as bioprocess engineering, also known as chemical engineering, because we create the sterile condition for the production of large number of desired microbes or the host cell. Now, there are three steps in genetically modifying organisms through which there are three steps through which we can genetically modify an organism. Number one is the identification of the DNA with desirable gene. This is a genome. Here's the gene of interest. First, I have to identify the DNA with the desirable gene of interest. Then only I will able to extract or take out the gene of interest. So I'll use the molecular scissor, which are the restriction enzyme. The restriction enzyme will cut, and we can easily take out the gene of interest. Now to incorporate this gene of interest into the host cell, I need a vector. I need a vehicle. For example, if I have to go somewhere from one place to another, I need a vehicle. It could be a bus, it could be a cycle, it could be a car. Similarly, the vector, they also act as a vehicle or a carrier. Now, these vectors, they have to carry the gene of interest into the host cell. So I have to cut this vector with a restriction enzyme so that they produce the sticky end. And this gene of, so that this gene of interest can combine or join with the vector. So this is the second step. That is the introduction of identified DNA into the host. Now I will introduce this recombinant vector or the recombinant DNA into the host cell. Third step is the maintenance of introduced DNA in the host and transfer of the DNA to its progeny. When this recombinant DNA is introduced or incorporated into the host cell, now as the host will divide or reproduce, this recombinant DNA will also transfer from the parent to the progeny. So its number increases. So Stanley Cohen and the Herbert Boyer, they are the scientists who are involved in the construction of the first recombinant DNA in 1972. Remember this year, it's very important that the first recombinant DNA molecule was constructed in year 1972, and the scientists are Cohen and Boyer. What they did, they take the plasmid, and this plasmid contains the antibiotic resistance gene. Okay. And they take this antibiotic resistance gene and incorporate it into the plasmid of another bacteria, which is Salmonella typhimurium. This is how they created the first recombinant DNA. This is a plasmid from Salmonella typhimurium. They incorporated this recombinant antibiotic resistant genes into the plasmid of Salmonella typhimurium. Now this become a recombinant DNA. And they incorporated this recombinant DNA into the host cell, which is E. coli. So the process of joining and inserting a foreign piece of DNA into the host organism to produce a new genetic combination is known as recombinant DNA technology. Because we are introducing the gene of interest or the foreign DNA into the host cell with the help of vectors. So it's lead to a formation of a recombinant DNA, and the technology is known as recombinant DNA technology. Now let's discuss the tools which are involved in recombinant DNA technology. Number one is a molecular scissor, and the function of the scissor is to cut, right? So the molecular scissor here are the restriction enzyme. So in year 1963, the two enzymes were responsible for restricting the growth of the bacteriophages. The one enzyme, it cut the DNA, and the function of other enzyme add the methyl group to the DNA. Okay, let me draw it. If this is we know that the function of a bacteriophage is to infect the bacteria. Okay, it's a bacteriophage is a virus, and it infects the bacteria. So if this is a bacteriophage, this is a viral DNA. And this is a host DNA. So this host, which is an E. coli, has two enzymes. One cut the DNA, and other one add the methyl group. Okay. And the second one, which add the methyl group. Okay. So the first one which cuts the DNA, this first enzyme, it cut the viral DNA. So the viral DNA has been destroyed by this enzyme, which is cutting the DNA. So the viral DNA cannot infect the host. So these enzymes, they're restricting the growth of the bacteriophage because they are not allowing the bacteriophage to grow or to multiply in the host cell. So the first enzyme which cut the DNA is the restriction enzyme. It was later known as restriction enzyme. But we know that the restriction enzyme cut the DNA, but this restriction enzyme is not cutting the DNA of the host cell because there's another enzyme is present in the host, which is DNA methylase. This enzyme adds the methyl group to the DNA of the host cell, because of which the host DNA get protected from the restriction enzyme. So restriction enzyme cannot act on the host DNA. Okay, I hope it is clear. Shishant, I hope it is clear. The first is the restriction enzyme, and the second, which add the methyl group to the DNA of the host cell, is DNA methylase. And restriction enzyme, they are further of two types. They're exonuclease and endonuclease. Exonuclease are the one which cut the DNA from the terminal end. And endonuclease are the one which cut the DNA within. Okay? Now there's a cloning vector. I told you vector is a vehicle. It acts as a vehicle or a carrier, which carry the gene of interest into the host cell. And the third one is the host cell. We require a competent host cell, so that when the vector containing the gene of interest is incorporated into the host cell, as the host reproduce or at the host divide, the number of these recombinant DNA also increases in the host cell, and we can get the desirable product. Next is a restriction enzyme. Restriction enzyme, there are two types. I told you it's exonuclease and endonuclease. Exonuclease will cut the DNA from the terminal end. If this is the DNA, it cut the DNA from the terminal end. And endonuclease cut the DNA within. Like it could cut here, it could cut here. Okay. And endonuclease, restriction endonuclease, they produces two type of ends. One is the sticky end, and another one is a blunt end. Another name for the sticky end are staggered end, overhanging ends, or cohesive ends. These are the alternative names of sticky ends. Another name for blunt end is flush end. And remember this, it's very important. The first restriction endonuclease is HINT2. S stand for Haemophilus and I stand for influenza. So the first restriction endonuclease is HINT2. Remember this, it's very important. And the restriction enzyme, they cannot cut like randomly in the DNA. They cut at a specific site, and these sites are known as the recognition sites. And these sites, they have a palindromic sequence. Palindromic sequence means, for example, if this is a DNA strand, it's having a polarity 5' to 3'. Having a sequence like A, G, T, T, C, A. Okay, hypothetically, I'm giving you the sequence. This is a second strand which is complementary. So here it will be T, C, A, A. Okay, just give me. For example, this is one strand and this is another strand. This is also having a A, G, T, T, C, A. So when I read it from 5 prime to 3 prime, it is pi A, G, T, T, C, A. If I read this strand also from 5 prime to 3 prime, it is A, G, T, T, C, A. So it is the palindromic sequence. But you have to read it in the same direction, like 5 prime to 3 prime. And let's come to the naming of restriction enzyme. Here's the example is given of E. coli. So E stands for genus, CO stands for species, R stands for strain, and I is the isolation order. Okay, because this is the first enzyme which is isolated from the strain RY13. So it is EcoR1. So 1 is the isolation order. Here it's a hint 2. 2 is the isolation order for hint. I told you that the endonucleases, restriction endonuclease, they produce two type of ends, the sticky ends and the blunt end. And remember one important point, that vector DNA and the foreign DNA, that is the gene of interest, should be cut with the same restriction enzyme. Because if they cut with the same restriction enzyme, that means they are going to produce the sticky ends. And we know that the DNA are complementary to each other, right? So we cut with the EcoR1, and each restriction enzyme, they have a specific recognition site. Okay, they will cut at a specific recognition site. And one more important point, that these restriction enzymes, they are going to cut both the strands of the DNA. They are going to cut both the strands of the DNA, and the cut will be made between the similar base pairs. You can see this EcoR1, here it is cutting between G and A, here it is also cutting between GA. Okay. Here also in the foreign DNA, the EcoR1 is cutting. Here also it is cutting between G and A. Here it is also cutting between G and A. Okay, so they are cutting between the similar base pair, and they're cutting both the strands of the DNA. Then only they are going to produce the sticky ends. And we know that DNA are complementary. So this AATTT, which is a sticky end, they will bind with the AATT, and they will form the recombinant DNA. And this reaction, it also requires an enzyme, which is a DNA ligase. DNA ligase, they act as a molecular glue. They help in the joining of the vector DNA and the foreign DNA. And I told you that every restriction enzyme, they have a specific recognition site, like ALU1, they have a recognition site at GC. Here it is also GC. These are the palindromic sequence. C. Here it is, if I'm reading it from 5 prime to 3 prime, the sequence is A, G, C, T. If I'm reading it, the second strand from the 5 prime to 3 prime, again the sequence is A, G, C, T. This is what we call palindromic sequence, okay? Remember this. You have to read this in the same direction, like 5 prime to 3 prime. Here you also have to read it in the same direction, 5 prime to 3 prime. Then is the palindromic sequence. And each restriction enzyme, whether it is ALU1, which is HEE3, which is BAM-H1, HIN3, EcoR1, they all have a different restriction sites. They will recognize a specific sequence. Okay, so you can see this HIN3 is cutting between AA and AA. EcoR1 is cutting between GA. Here it is also cutting between GA, right? And today, there are more than 900 restriction enzyme has been recognized from over 230 bacterial strains. And each restriction enzyme, they have a different recognition site. So this is a foreign DNA. I expose this foreign DNA with the restriction enzyme. So restriction enzyme, they will act as a molecular scissor. They will cut the foreign DNA, right? And the vector is also exposed with the same restriction enzyme. So the restriction enzyme, they will recognize the site in the vector and they will cut the vector also. I'm using the same restriction enzyme. Okay, remember this. And this foreign DNA will get incorporated into the vector. For this, we require an enzyme which is a ligase, DNA ligase. And DNA ligase, they act as a molecular glue to join the foreign DNA and the vector DNA. Now this vector DNA, which is containing the foreign gene or the gene of interest, will be incorporated into the host cell. And the process is known as transformation. Now, as the host cell divide or reproduce, this recombinant DNA will also increase in number. Next one is the agarose gel electrophoresis. Agarose, because we are using a gel, agarose, and electrophoresis because we are using the electric field. Once you have subjected or exposed the genome with the restriction enzyme, now you have to separate the fragments of the DNA. For that, you require a gel electrophoresis. So main function of gel electrophoresis is to separate the fragments of the DNA on the basis of their size. Remember this, on the basis of their size. We know that DNA are negatively charged. So this is a cathode, and cathode is negative and anode is positive. So DNA will always travel because they're negatively charged, so they will move towards the positive charge, which is anode. Here's anode in positive. So DNA will move in this direction. And the fragments of the DNA, which are smaller in size, will travel the maximum distance. You can see that this fragment is smaller in size, so it's covering the maximum distance, and the fragment which are larger in size will cover the smaller distance. So on the basis of the size, you can separate these fragments. And these fragments are the DNA fragments, they are stained with ethidium bromide. So when they are exposed to UV film or the UV radiation, they give orange color. Next one is the cloning vector. As I told you that the vector, they are the vehicle, they are a carrier for introducing the segment of the foreign DNA into the suitable host. And the example of the vector are plasmid and bacteriophage. Plasmid is the most common vector. And there's certain characteristics about the vector. The vector should be small in size so that we can easily handle it. Second, a vector should have the origin of replication. We know that the replication cannot occur at any random place. There should be a specific site or the sequence where the replication can initiate. And these sequences are known as origin of replication. So the foreign DNA or the gene of interest, which is linked with the origin of replication, can replicate in the host, and this origin of replication also controls the copy number of the DNA or the linked DNA. And the vector should contain a selectable marker. Selectable markers, they help in identifying the non-transformant and the transformant host. Like it helps you in identifying or eliminating the non-transformant host, because only the transformant host will able to grow. So the vector should have a selectable marker, and the most common example of selectable marker is the antibiotic resistance gene and the cloning sites. Each vector, they have a cloning site, the site where the restriction enzyme will cut. And remember, a good vector is the one which has a restriction sites for a variety of enzymes. Like they have a cloning sites for a variety of restriction enzymes. For example, you can see in the figure, this is a diagram of a artificial plasmid, or artificial vector, PBR322, where P is the for plasmid, BR is the name of the scientist, that is Bolivar and Rodriguez, and 322 is the number. Like you people have your roll number, right? You have the roll number in the class. Similarly, this vector also has a number which is 322. Remember this diagram, it's very, very important. Remember this diagram as it is. Remember the where the eco-R1 site are present, where the site for PVU1 is present, where the site for PST1 is present, BAMH1, SAL1. Remember these sites, CLAY1, HIN3. These are very important. And remember, in the same sequence, okay? This is also important. You have to remember in the same sequence. And the two selectable markers are present. There's tetracycline and there's ampicillin resistance gene. This vector contains origin of replication. And one more important point, that these restriction enzymes, they have their cloning site in the selectable marker. Like BAM-H1 and SAL1, they are having their restriction sites or their recognition sites in the tetracycline resistant gene, which is a selectable marker. So the cloning sites, they can be present within the selectable marker. Remember this point. And remember that tetracycline resistance gene, they contains BAM-H1 and SAL1 restriction sites. Similarly, the ampicillin resistant gene, it contains the restriction sites for PVU1 and PST1. Okay, and a good vector is one which is having a restriction sites for the variety of, which are having a cloning site for the variety of the restriction enzyme, so that I can use any of the enzyme. For example, I can use PST1 to cut this vector. I can use BAM-H1. I can use SAL1 because I have an option to keep that. I can use any of the restriction enzyme to cut this vector. But if the vector is having a more cloning site for a single restriction enzyme, like I'm having a single restriction enzyme, which is EcoR1. Okay. And this EcoR1 is having a multiple cloning site in this vector. It can cut here, it can cut here, it can cut here, and cut here, because the multiple cloning sites are present. Then it will break the vector. It will destroy the vector. We cannot use this vector. So a good vector should have a recognition sites for a variety of restriction enzyme, instead of having a multiple cloning site for a single restriction enzyme. Okay. This diagram is very important. Remember this. Now let's take an example. This is a vector. This is the ampicillin resistant gene, and this is the tetracycline resistant gene. These are the selectable marker. Okay. And this is a gene of interest. And I expose this vector with the restriction enzyme. Now there's a three possibility that the gene of interest can be incorporated into the tetracycline resistant gene, right? Or it does not incorporate into the vector. So there's two possibilities. Here the ampicillin and tetracycline resistant genes are intact because the gene of interest has not incorporated or not inserted. And here the gene of interest has incorporated. Okay, so this is non-recombinant. We always use the term recombinant and non-recombinant for the vector. Remember this. For the host, we use the term transformant or non-transformant. And this is recombinant because the gene of interest has entered or inserted into the vector. Okay, I'm getting the two conditions. Now there are further three possibilities. That either the host cell, which is the E. coli, can contain the non-recombinant vector or the recombinant vector, or even does not contain, or it does not get in any of the vector. Okay, so this E. coli cell, it does not have any. It does not have the non-recombinant vector or not even the recombinant vector. There's a second possibility, and we call these host cell as non-transformed. There's a second possibility that this E. coli cell, host cell, contains this non-recombinant vector. Okay, And there's a third possibility that the E. coli cell can have this recombinant vector. So then the transformed cell or the host cell which is containing the non-recombinant vector will be called as transformed with non-recombinant vector. And the other host cell is the transformed with recombinant vector. Okay. Now you know that the non-recombinant vector, the ampicillin and tetracycline genes, they are intact. That means the host cell which is containing this non-recombinant vector will able to grow in the ampicillin medium also and in tetracycline medium also, because they have the ampicillin resistant genes and they have tetracycline resistant gene. Okay. So they can grow in both the medium. But the one which is having a recombinant vector, the one which is having the recombinant vector, that means the gene of interest has incorporated into the tetracycline resistance gene. Okay. So because of the insertion of the foreign DNA or the gene of interest into the tetracycline resistant gene, it leads to the inactivation of the gene. So tetracycline resistant genes will become inactivated. So this host cell will not be able to grow in the medium which contains tetracycline, because the tetracycline resistant gene has become inactivated due to the insertion of this foreign DNA into the tetracycline resistant gene site. Okay, but this transform cell will be able to grow in the medium which is containing the ampicillin, because it has the intact ampicillin resistant gene. Okay, I hope it is clear to everyone that what's the difference between non-recombinant and recombinant vector, and the difference between non-transformed, transformed with non-recombinant vector, or transformed with recombinant vector. There's another example. See, this is a plasmid vector, and this plasmid vector, this blue portion, light blue portion, represent a gene which is LAGZ. Okay, and this LAGZ gene is code for a enzyme which is beta galactosidase. So when this LAGZ gene is intact, it produces a beta galactosidase enzyme. This beta galactosidase enzyme will act on a substrate which is X-gal. This is a substrate which is present in the medium. This X-gal will convert into a product, and this product is blue in color. Okay. Now discuss this image. So here's this a plasmid which is which acting as a vector. It containing an intact LAGZ gene, right? This small blue color, light blue color is the LAGZ gene, and this is the foreign DNA, red color. So now there are three possibilities. Either the foreign DNA is not inserted into the LAGZ gene. Okay, if it is not inserted, that means LAGZ is intact. And this LAGZ will produce beta galactosidase enzyme, and this beta galactosidase will act on the X-gal which is present in the medium, and it will produce a product which is blue in color. So we are getting a blue colonies, right? Second possibility could be that this foreign DNA has inserted into the site other than the LAGZ. You can see this. It has inserted into the site other than the LAGZ. So that means the LAGZ is intact. It will again synthesize beta galactosidase, and beta galactosidase will act on the X-gal substrate, produce a product which is blue in color, and again we are getting a blue colonies, right? But the third possibility could be that this foreign DNA has inserted into or incorporated in within the LAGZ. So if this gene of interest or the foreign DNA has inserted within the LAGZ, that means the LAGZ will become inactive. Okay, now this LAGZ will become inactive, so it won't be able to synthesize this beta galactosidase enzyme. If this beta galactosidase enzyme is not formed, now it will not be able to act on the substrate which is X-gal present in the medium. So no blue product will be formed and the colonies, they appear white in color. Okay, I hope it is clear to everyone that why we are getting a blue colonies or the white colonies. So the blue colonies will carry the non-recombinant and the white colonies will carry the recombinant vector. There are two types of vector are present in plants and animals. For plants, we use agrobacterium or the TI, Agrobacterium tumefacium. It's a pathogen which causes the crown gall disease or the tumor disease in dicot plant because they transfer the DNA which is known as a tDNA. So for plants, we use a TI plasmid. TI plasmid, they contain this virulent lesion, okay? And this virulent region is responsible for causing the crown gall disease or the tumor disease in the host cell. So if we remove this virulence region, then this plasmid would be disarmed because we have modified this vector. And this plasmid then can be used as a cloning vector for the plant. For animal, we use a cloning vector as a retroviral, but we cannot use it like directly. We have to modify it. We have to remove this virulence region so it does not cause the disease in the host cell. For that, we have to first modify the vector. Next one is the competent host. First treatment is the Cl2 treatment. We know that the DNA are negatively charged, right? And this calcium chloride, the calcium is positively charged. It's a divalent cation. So this calcium, this surrounds the DNA. When we treat the host cell with the calcium chloride. Now we give the host cell pre-treatment. First ice, then heat at 42 degrees Celsius and again ice. After this treatment of ice, heat and again ice, it creates the pores in the membrane of the host cell. When the pores are created, the DNA, which is surrounded by the calcium, move into the host cell. So we are making the host cell competent for the foreign DNA. Next one is microinjection. Microinjection is done only for the animal cell, where we injecting the recombinant DNA directly into the nucleus of the animal cell or the host cell. Ballistic or the gene gun, this is used for plant cell, where the gold or tungsten particles, they are coated with DNA and they are bombarded with the high velocity to the target cell. This some pathogen vector are TI plasmid or the retrovirus. We disarm those vector. We have to modify those vector before using. Now, first is the isolation of a specific genetic material. If we have to isolate the genetic material from DNA, we know that if we have to isolate the genetic material from different organisms like bacteria, plant cell, and fungi, we have to treat these organisms with their certain cell wall degrading enzymes. Like bacteria, they have a peptidoglycan cell wall. So I have to subject or expose the bacteria with lysozyme to degrade the cell wall of the bacteria. Second one is the plant cell. The plant cell, they have a cell wall cellulose. So I have to expose the plant cell with cellulase enzyme. Fungi, they have chitin cell wall. So I have to expose the fungi with chitinase enzyme to degrade their cell wall. And we know that the cell extract, apart from DNA, they also contain a lot of things, like they contain RNA, they contain lipid, they contain proteins, right? So I have to treat this cell extract with RNase enzyme to degrade the RNA, lipase to degrade the lipid, proteinase to degrade the protein, so that I can only get DNA. And now this purified DNA is ultimately precipitated out by using the chilled ethanol. In this diagram, you can see This is the precipitate DNA. And we are taking out this DNA with the help of spool pick, and the process of taking out the precipitate DNA in the intact state, state is known as a spooling. So now let's discuss the process of recombinant DNA technology. The first, this is, for example, this is a genome, and this genome has a lot of genes. And I have to take out only the gene of interest. First, I have to expose this genome with a restriction enzyme, right? Restriction enzyme will cut this genome and we'll get the fragments of DNA. Now I have to separate these fragments of DNA, so I have to carry out the gel electrophoresis. If I get my gene of interest, now I have to amplify because it's a very small amount. I have to multiply, or I have to increase the I have to increase the amount of gene of interest. So I have to subject the gene of interest to PCR, which is a polymerase chain reaction, to increase the amount of the gene of interest, because I require this gene of interest in a very large quantity. So remember the full form of PCR. It's very important. PCR is a polymerase chain reaction. We know that for the separating the DNA, the fragments of the DNA, we use an enzyme, and the enzyme is known as helicase, right? But here in PCR, we are not using the helicase enzyme. Here, we are separating the two strands of the DNA with high temperature. And at high temperature, the two strands of the DNA, they get separated. The hydrogen bond, which is present between the nitrogenous bases, they get break, and the two strands of the DNA, they get separated. Now, after the separation, the second step, so the first step is denaturation, because the two strands of the DNA, they get separated, and the process is known as denaturation. Second process is primer, or second step is primer annealing. The primer, they will bind to the respective strands of the DNA. The function of the primer is to provide a free prime OH group, so that the incoming nucleotide can add to the primer. So this step where the primers are added to the strands of the DNA, the step is known as primer annealing. And now the third step is extension. For extension, that means we have to add the nucleotide to the primer to form a long chain. For that, we require DNA polymerase, right? But here, the special type of DNA polymerase is present, which is known as a tag polymerase, because this reaction is taking place at a very high temperature. So we require an enzyme which is thermostable. So tag polymerase is a special type of DNA polymerase, and it is thermostable. And this tag polymerase is isolated from an enzyme which is Thermus aquaticus. So this tag polymerase, which is a special type of DNA polymerase, is isolated from an organism, and the name of the organism is the Thermus aquaticus. Remember the name of the organism from which the tag polymerase is isolated. It's very important. Tag polymerase is thermostable, so we are using tag polymerase in PCR. So main function of tag polymerase is in polymerization, or keep on adding the nucleotide to the primer. So in 30 cycles, 1 billion times the product is amplified. And remember the step. The first step is denaturation, the second step is primer annealing, and the third step is extension. Okay, remember the step. These are very important. Many times the question came in need from PCR. Once the gene of interest is amplified, or its number is increasing, now we have to insert the gene of interest into the vector. Now this vector will carry the gene of interest into the host cell. Now the host cell, they contain the gene of interest. Now we have to grow this host cell in a very large quantity because we required a large amount of product, because our desirable aim is to get the product. So we have to culture this host in a very large quantity. For that, we require bioreactors. So there are two types of bioreactors are present. There's simple stirred bioreactor and there's a sparged bioreactors. So a bioreactor should have a good aeration, should have a proper acid-base control, should have a proper temperature control, should have a proper form breakers blade. Okay, these are the characteristics of the bioreactors. This is the simple stirred bioreactor and this is the sparged bioreactor. You can see this, in sparged, there's a oxygen tube is present, which is supplying the oxygen. Next one is a downstream processing. So downstream processing means purification and extraction of the product, right? And before marketing, the preservatives are added. So this is known as downstreaming processing. Purification and extraction of product. Second one is addition of preservative. So two steps are done in biostreaming processing. Number one is purification and extraction of the product. Second one is addition of preservative to the product before they are marketed. Now let's discuss the question. Question 1 says, in a cell, the separation of DNA strands is brought about by the enzyme DNA helicase, whereas in PCR, the separation of DNA strands is due to, option 1 is two set of primers, option 2 is tag DNA polymerase, option 3 is deoxynucleotides, and option 4 is high temperature. What do you think? What will be the correct option? Very good, Shushant. Very good, Veena. So normally, if we have to separate the two strands of DNA, we generally use DNA helicase, right? We use helicase in replication to form the replication fork. We also use helicase in transcription to form a transcriptional bubble, right? Because we have to separate the strands of DNA. But in case of PCR, and remember the full form of PCR is polymerase chain reaction. So in PCR, the first step is denaturation, where high temperature is given. At this step, approximately 90 to 95 degree Celsius temperature is given, and at which the two strands of the DNA, they get separated. Now the hydrogen bond between the two strands, they get break, and the two strands are separated. This is the first step. Second step is annealing. Annealing means joining. So primer will anneal to the single strands of the DNA. Third step is extension. Here, tag polymerase play an important role because the function of tag polymerase is polymerization. So tag polymerase will keep on added the nucleotide and they form a long chain. This is how the amplification is done. So the correct option is fourth. I hope it is clear to everyone. Moving next is, which of the following is the correct sequence of steps in PCR, which is polymerase chain reaction? It's very easy. Quickly answer everybody what you think, what will be the correct option. Sushant asking about the correct sequence. Just now I explained the PCR. Dhruvi, Priyadarshini, what do you think, what will be the correct option? Very good, Veena. Very good, Sushant. So PCR include 3 steps. Number 1 is denaturation, separation of the DNA fragment due to high temperature. Second one is annealing. We also call it as primer annealing, where the primer joined to the strands of the DNA. Third one is extension. Once the primer joined, now the Taq polymerase, they will keep on adding the nucleotide to the primer. So the first is denaturation, second one is annealing, third is extension. So the correct option is 1. I hope it is clear to everyone. They're asking about the correct sequence. Now moving next. In the following palindromic base sequence of DNA, which one can be cut easily by a particular restriction enzyme? Quickly answer what you think what will be the correct option. Try to recall what I told you about the palindromic sequence, 5' to 3', then you can easily answer this question. Very good, Veena. Very good, Sushant. So in option 1, if you see from 5, if I talk about from 5' to 3', then it's GAATTC. If I read the sequence from the 5' to 3', it's GAATTC. So it's a palindromic sequence. Second says 5' to 3' is CTCAGT. From 5' it's ACTGAG. No. Third says GTATTC. GAATAC. No. GATACT. AGTATC. No. So the correct option is 1. This is the correct palindromic sequence. I hope it is clear to everyone. Always read from 5' to 3'. Like from in this direction and from this direction. Question 4 says, select the correct statement from the following. Number 1 is gel electrophoresis is used for amplification of a DNA fragment. 2 says the polymerase enzyme joined the gene of interest and the vector DNA. Third is restriction enzyme digestion are performed by incubating purified DNA molecule with the restriction enzyme of optimum condition. Fourth is PCR is used for isolation and separation of the gene of interest. What do you think? What will be the correct option? You have to find out the correct statement. Statement 1 is saying that gel electrophoresis is used for amplification of DNA fragment. No, for amplification, we require PCR. Amplification means for multiplication. So we require PCR, not gel electrophoresis. We require gel electrophoresis to separate the fragments of the DNA. Option 2 says that polymerase enzyme join the gene of interest and the vector. No, this is the function of DNA ligase. DNA ligase join the gene of interest, that is a foreign DNA with the vector DNA. Polymerase का ये function नहीं होता, it's a function of DNA ligase. Very good, Veena. Third says the restriction enzyme digestion are performed by incubating purified DNA molecules with the restriction enzyme of optimum condition. Yes, absolutely true. Fourth says that PCR is used for isolation and separation of gene of interest. No, PCR are used for amplification of gene of interest. So the correct option is 3. I hope it is clear to everyone. Now we are winding up the class. Thank you all for your participation. I hope this lecture was informative for you. Thank you all and see you in the next class. Have a nice day.